Okay, let's continue to talk about the neurons, the cell and the membranes and more importantly right now, the ion channels that are in the membranes, whether the membrane be covering the axon, be covering the nerve cell body, or be covering the dendrites, because we may have different ion channels according to where these are at. So let's talk about what do we mean by these ion channels, which they are always integral proteins, meaning they extend from one side of the membrane through the other side, and they have pores in them that allow ions to obviously pass into or out of the cell. Okay, here again we see an example of our phospholipid bilayer. Remember, every cell is covered by that cell membrane made primarily of phospholipid bilayer, and of course all these are proteins. Specifically, these proteins are ion channels, meaning they are there to allow ions to pass into or out of the cell. But what we want to start establishing is what are the categories of these channels. And so there are some channels that are always open. We're going to call those leak channels. There are some channels that will open and close according to different phenomenon, like a voltage change or a chemical attaching to them, or even a mechanical stimulus. And so we would call again those voltage gated channels or chemically gated channels or mechanically gated channels. And then we're going to see that some of those specific channels, especially sodium voltage channels, actually have kind of a mechanism, two different things. It has a voltage portion and a timed gate. And so we're going to look at that phenomenon a little bit later. Okay, each of these channels have certain characteristics. They are selective. In other words, they will only allow certain things to pass through them. They're either gated or non-gated. Now again, gated means that they basically have the ability to close, and so we can see this is non-gated here, or it could be representing non-gated, meaning that it is always open. We call those leak channels, but the rest of these are gated, and that means that they have the ability to open and close due to different stimulus. And again, the stimuli is either we have a neurotransmitter attaching to them, like a key opening up a door. We have a voltage change that can open them up, or we have a mechanical change that can open them up. So they're gated or non-gated. And then regionally located, where would we find the different types of channels? What kind of channel would I find here? Well, I'm probably not going to find a keyed channel or a chemically gated channel here. Why? Because the chemical is the neurotransmitter. This is the conductive area. So where are my receptive areas? Where are the areas where I would receive neurotransmitters? Well, that would be my dendrites and my nerve cell body. So I would expect the channels here to be the ones that could receive the neurotransmitter. So that would be my chemically gated channels. So again, we're going to show that later. But again, I'm going to have different channels in different areas, even though there are a few of the channels that I may find everywhere, especially these leak channels. But we'll talk about why that is. So again, channels are selective. This anion here, whatever it represents, could be a protein, could be another large molecule, is too big to pass through these channels, so it's not going to go through it. Again, they're basically the passageways are basically based on either charge of the ion, because negatively charged something's not necessarily going to be able to go through something that is equipped to allow positive ions to flow through it. And of course, size makes a big difference. If sodium has a certain size, there are some channels that will allow both sodium and potassium to pass, but there are some channels that would only allow sodium to pass because of its size difference with potassium. Okay, so again, the channels can be gated or non-gated. Okay, if they're non-gated, they are leak channels, meaning that they are always open, meaning that basically what will determine what goes through them will be the size, the charge, and do I have a higher concentration on one side than the other? If I do, it'll go from the high concentration to the lesser concentration. Okay, and in this case, I believe this is supposed to be potassium, so it would be going in this direction. All right, but gated channels then, again, as I've mentioned several times, meaning that they can open and close due to different types of stimulus. Okay, here we have an example of a gated channel that happens to be a voltage gated channel. So the only way it will open up is if I can change the voltage on the inside. Well, what would change the voltage on the inside? because if I have a certain amount of ions outside and inside, I get a voltage difference here. I call that my membrane potential. When I only have leak channels open, then I maintain something called a resting membrane potential. But what if for some reason I all of a sudden have a certain rush of ions into the cell? Then that would certainly change the potential, and it could change the potential to a specific voltage reading called threshold. And once we reach that specific voltage reading, read in millivolts of threshold, threshold is what actually causes our voltage channels to open up. Therefore, more ions can rush in. So voltage channels are opened up by a change in voltage. A change in voltage comes about due to ions coming into the cells. Well, how do I let the ions into the cells that would change the voltage and open up my voltage-gated channels? Well, that's where my chemically gated channels come in. These channels are not opened up by voltage. They're not leak channels. They're only open up when I put a key in the doorway. All right, so the key here happens to be acetylcholine. So if I release a neurotransmitter, I open up this door, ions come through. When these ions come through, then it changes the voltage, and if enough of these ions come through where I change the voltage to threshold, then I open up my voltage-gated channels. All right, but I also, that is if we're talking about an excitatory situation, and because if I don't reach threshold, then that would be an inhibitory situation. So if I have neurotransmitters that open up doorways that allow positive ions to flow in, that is definitely an excitatory situation. But if my neurotransmitter opens up a doorway that allows positive ions to leave, then that's an inhibitory situation. Or if I have a neurotransmitter such as GABA that actually opens up a doorway and allows a negative ion, and in this case what GABA does, it allows chloride to come in, then that actually creates a negative situation in here, which will not allow me to reach that magic number of threshold. Therefore, my voltage channels will not open up. So again, if these are chemically gated channels, the neurotransmitters are the chemicals. Either they open up the doorway and allow positive ions to come in. That leads towards excitement, towards opening up the voltage channels, or the neurotransmitter goes to a receptor that allows positive ions to leave. That's going to inhibit what's going on inside. Or I will have a neurotransmitter that will actually allow negative ions to come in, which will also inhibit the situation. Here we see acetylcholine opening up this doorway. Sodium is rushing in. So since sodium was positive rushing in, then it's going to change my voltage inside towards a more positive number. If I get enough sodium in or reach threshold, then I start opening up my voltage gates. But here we see GABA being the key to my doorway, opening it up and allowing a negative ion, such as chloride, to come in. Therefore, if negative ions come in, prevents me from reaching threshold, and therefore my voltage gates will not open up. Okay, now very importantly, where am I going to find these different types of doorways or channels? I like to call them doorways just to kind of relate it to something you're familiar with. Okay, notice I've got non-gated or leak channels. Where would I find these? I've got chemically gated channels. Where would I find these? And I've got voltage gated channels. Where would I find these? Well, understanding what these lead to will help us understand what is going on here. So the first thing I want to say is that non-gated channels are responsible for what's called the resting membrane potential. That voltage we can measure anywhere inside the membrane, anywhere in the cell when nothing else is happening. When we have the positive charge on the outside compared to the relatively negative charge on the inside of the membrane, that's being maintained by the number of potassium and sodium channels, especially the potassium channels. So since I can measure a resting membrane potential anywhere, then that means what's causing that resting membrane potential is the non-gated channels. Therefore, I would find non-gated channels everywhere in the neuron. Okay, next then, my chemically gated channels. Well, my chemically gated channels have to be where I am going to be receiving my neurotransmitters. So where is our receptive areas? Well, our receptive areas are our our dendrites and our nerve cell body. Well, the reason why they're receptive areas is because the doorways have keyholes in them, and the chemically gated channel, the chemical is the neurotransmitter. So I wouldn't expect to find chemically gated channels here in my axon. Now, there's an exception to that at the end of the axon, but as far as the majority of the axon, I wouldn't expect to have chemically gated channels here because I'm not releasing neurotransmitters there. So it wouldn't do me any good. Plus my voltage gated channels, that's what I'm going to find starting at my axon hillock and throughout the rest of my axon. Or if this axon's myelinated, then of course I don't need channels under the myelin, but I do need channels in those nodes of Ranvier that we talked about. Because these channels, these voltage gated channels, once I reach threshold, once I reach this voltage due to what happened at the chemically gated channels, then once the sodium rushes in, then it causes the next voltage gated channel to open up, which then once sodium rushes in at that point, it causes the next ones to open up. So these are going to be found from the axon hillock throughout the rest of the axon, but they can only be opened up once we reach our voltage, once we reach our threshold voltage at the axon hillock. And of course, it's the chemically gated channels that allow us to either reach threshold by excitatory methods or prevent reaching threshold by inhibitory methods.
On dendrites, the cell body, and the axon. For the most part, chemically gated channels are located on the dendrites and cell body of the neuron. For the most part, voltage-gated channels are found on the axon hillock, all along unmyelinated axons, and at the nodes of Ranvier in myelinated axons.
Okay, so again, I wanted you to see where these are found, which is just a very important concept that you do need to grasp and you do need to remember. Now, not only do we need to understand where we would find these different channels, as I alluded to earlier, we need to understand what are these channels causing in the neuron. So what are the potentials that they are causing? Well, let's start down here. The action potential, which is the signal starting at the axon hillock and moving all the way through or moving at the nodes, is caused by voltage-gated channels. The resting potential, what's going on in the cell when we're not receiving or sending information, so it's going on all the time, but it's also going on when nothing else is happening, is caused by the non-gated potential. So what's happening at the synapses, where we are releasing our neurotransmitter, is called the synaptic potential, also called the graded potential. We'll explain why it's called both of those later. But the synaptic potential or graded potential is basically what's happening at the synapses where we have the chemically gated channels.
Non-gated channels are responsible for the resting membrane potential. We will learn about the resting membrane potential in the next section of this module. Chemically gated channels are responsible for synaptic potentials, the incoming signals to the neuron. Voltage-gated channels are responsible for generation and propagation of the action potential, the outgoing signal from the neuron. We will learn about the action potential in the last section of this module.